I want to dive in and talk more about the material. So let's do that. I want to finish up with a couple of things today that I didn't get finished up on last time. And they actually relate to the material that we'll talk about today for the mechanism of enzyme action. So the first of these is a... well, actually, let's see. I see I didn't finish up here Lineweaver Burk, so maybe I should do that. When I talked about non-competitive last time, one of the things about non-competitive inhibition that, as I said, you may find is not intuitive to you, and that is that the Km for non-competitive inhibition remains the same. It remains unchanged. The Vmax, however, for competitive inhibition decreases. And the reason for that is because when you do non-competitive inhibition, you don't have varying amounts of inhibited enzyme. In competitive inhibition, the amount of enzyme that's inhibited is a function of the substrate concentration. That's not the case in non-competitive inhibition. In non-competitive inhibition, you have a fixed amount of enzyme that's inhibited in every reaction. Doesn't matter what the substrate concentration is. And as a consequence of that, it's the same as doing a reaction, or doing all the reactions, with less enzyme and no inhibitor. Okay? If I said that every reaction I did was going to have 25% of the enzyme inhibited, I would get exactly the same results as if I had done the same set of reactions with only 75% of the enzyme. Okay? Because that fixed amount of inhibition, all right, is always reducing the amount of enzyme by the same amount. Well, what I hope I pounded into your heads was that Vmax is not a constant. It's a function of the amount of enzyme that you use. So if all the non-competitive inhibition reactions that you do for a given set all have the same amount of enzyme, they are going to have the same Vmax. And since it's a lower amount than the uninhibited reaction, then you're going to have a lower Vmax for non-competitive. Similarly, if you think about it, since I would have the same amount of enzyme in every reaction, that same amount of enzyme is going to have exactly the same affinity for the substrate in each reaction, so the Km does not change. Now, if you find those concepts difficult to understand, and sometimes people do, come see me, and I'll be happy to explain them to you in a little bit different terms. Okay. Well, on Lineweaver Burk, this is what a non-competitive inhibition looks like compared to no inhibition. All right? And this actually, students get a little confused on to start with, but I'll take you through it. We remember that for non-competitive inhibition, the Km is the same, which means that as it is for an uninhibited reaction, which means that minus 1 over Km will be the same. And so these two lines cross at minus 1 over Km on the x-axis. Okay? You'll notice that the non-competitive inhibitor moves higher on the y-axis, 1 over Vmax, than the uninhibited reaction. And sometimes students find that confusing. But I remind you that we're plotting the reciprocal of the velocity on the y-axis. So since the Vmax of a non-competitive inhibition reaction is lower, 1 over Vmax will be higher. Okay? And that's why you see that looking like that. Okay, so that's the two types of inhibition that I talk about in the class, competitive and non-competitive. We won't talk about uncompetitive, which is conceptually quite a bit more difficult to understand, and I don't think it really helps us here to go through that. I do want to talk about other ways that we can, in fact, inhibit, and in fact, specifically inhibit and destroy enzyme function. Everything I've talked about so far relates to non-covalent binding of inhibitor to enzyme. Non-covalent binding. I mean, it can come on, it can come off, it can come on, it can come off, right? The next two things I want to talk about are covalent ways, covalent bindings that inhibit an enzyme. Okay? One of these is fairly non-specific, and it involves modification of an amino acid. This particular amino acid, if it is in the active site of the enzyme, will play havoc with the enzyme's ability to catalyze And playing havoc basically means it will stop it dead in its tracks. This inhibition results from the chemical modification of the side chain of serine. And it's relevant to what I'll be talking about later today because I'm going to be talking about what are known as serine proteases. That is, proteases that have a serine in their active site. This particular chemical is called DIPF, and it has a much longer name, but you can call it DIPF. In fact, I don't even remember the name of the structure off the top of the molecule off the top of my head. But this molecule is specific for the side chains of serine. It's not specific for active sites. It's not specific for enzymes. It's specific for the the side chain of serines. All right? So any side chain of serine in any enzyme can be bound covalently by this DIPF molecule. Now, we'll see today that the side chain of serine plays an essential role in serine proteases. If you destroy it by converting that OH into this monstrosity over here, the enzyme can no longer function. So this is an example of a non-specific covalent inhibition. It's non-specific in the sense that it's not specific for any given enzyme. It'll work on any serine in any enzyme. Now, if the enzyme in question does not have a serine at the active site, it's entirely possible to treat an enzyme with this compound and have the enzyme still be active. It's possible. Okay? But if there's a serine in the active site, the enzyme will almost always be inactivated. Another way to covalently inhibit an enzyme is through something I mentioned in the song that we sang on Wednesday of last week, and it's known as suicide inhibition. Okay? One example, and it's actually mentioned in the song, is this is the inhibition of a critical enzyme in bacteria by the by the compound penicillin. So we all know that penicillin is an antibiotic, and the way that penicillin works is by covalently and specifically binding to the active site of an enzyme that bacteria need to make cell walls. Now, you can imagine when you covalently bind to an enzyme, the enzyme isn't going to recover from that. Okay? That's in contrast to some compound that goes on non-covalently and comes on, comes off, comes on, comes off. This guy, once it goes on, stays on. And penicillin has a structure such that it is recognized by the active site of this enzyme. So this is a specific inhibitor, and a specific inhibitor that covalently binds to the active site. A specific inhibitor that covalently binds to the active site is known as a suicide inhibitor. The enzyme commits suicide by binding to this molecule. There are other examples besides penicillin, but penicillin is one of the best examples I can give you for that. The enzyme that does that is known as a transpeptidase, a glycopeptide transpeptidase, and that enzyme is important, as they say, for building bacterial cell walls. So if the bacterium can't make its cell wall, of course, it's not going to be able to divide and do its thing. So the bacterium essentially dies from that. One other, actually, no, I won't talk about the other as well. I'll leave that be for right now. Okay, so that's the end of what I want to say about enzyme inhibition. Questions about that? Everybody's still dazed by the exam. Question? Yeah, back here. I'm sorry, can you repeat your question? Okay, so the question is, if this DIPF treats an enzyme and it binds to serine that's not in the active site, will it inhibit the enzyme? Well, if you think about this, you don't exactly know. You can't exactly tell. Because why? Because protein structure is essential for function. And so for some enzymes, we might imagine that serine might be playing some important role in structure, and by binding to the side chain of serine, we might be disrupting that structure, and the enzyme might lose function. In other enzymes, serine, a particular serine might not have that essential role and might have no effect. So there's no rule one way or the other. But you can say that if it's in the active site, it almost certainly will inhibit it. Okay? There's another question? Yeah? No. Question is, are non-specific and specific suicide inhibitors both called suicide inhibitors? No. Only the specific inhibitors, that is those that are recognized specifically at the active site by a specific enzyme. So this penicillin, for example, isn't going to bind to a wide variety of enzymes. It's going to bind to enzymes like the transpeptidase that have an active site that can bind to it specifically. Okay? DIPF is a relatively small molecule. Okay? It's a relatively small molecule. It can fit into an enzyme in a lot of crevices very easily. And that helps it to bind to many of the serines that are found in a given enzyme. Other questions? Okay. Well, we're going to turn to what I will admit is one of my least favorite topics. Okay. You say, you're supposed to like everything, Kevin, right? But I'm not a big fan of mechanism, but I think mechanism is important. So I spend some time on mechanism, but I probably don't spend as much time on mechanism as many professors in biochemistry do. Okay? I like to give you the essentials of understanding mechanism without overwhelming you with this goes to this, goes to this, goes to this. You will see some of that today. And if I don't finish it today, you'll see some of that on Wednesday as well. Okay? So I try to be merciful in my discussion of mechanisms. I start the description of mechanism by talking about the general way in which proteases work. Now, we talked about proteases. Proteases are, of course, enzymes that break peptide bonds within other proteins. And they do this via a hydrolysis reaction. And whenever you hear the word hydrolysis, it means water is involved. So you can see water being involved on the left side of this molecule. And the result of this catalysis is that a peptide bond is broken. There's a peptide bond. There's what it looks like after it's been broken. And the proteases catalyze that process. We're going to see there are several different classes of proteases. We're going to focus mostly on what are called the serine proteases. Okay. If we look at the enzyme known as chymotrypsin, you may remember that chymotrypsin recognizes and cuts adjacent to several different amino acids within a protein. Here are two of the different amino acids recognized by chymotrypsin. And you recall that it cuts on the carboxyl side of these two in addition to some other amino acids as well. Here is where the cut would occur, and here is where the cut would occur, again, breaking on peptide bonds. Chymotrypsin is an enzyme that's been studied a lot, a heck of a lot. Okay? So the first studies of chymotrypsin to reveal its mechanism showed that, in fact, there was a serine that played a critical role in its active site, and that could be demonstrated quite easily by treating the enzyme with DIPF that we've seen before. And as a result of the treatment with DIPF, the enzyme was stopped dead in the water. Now, it doesn't prove that a serine is in the active site, but it's consistent with a serine being in the active site. Okay? We'll see that, in fact, it is active in the active site. Okay? The way in which this enzyme has been studied has been through the use of a compound that changes color upon being acted upon by chymotrypsin. So I like to say that biochemists are lazy people because I'm a biochemist and I'm a lazy person. All right? Actually, I'm not a lazy person, but I can play one on TV, I suppose. Detecting breakage of a peptide bond is not something that one does readily. If one were to study that in a laboratory, you would have to, for example, run gels and do analyses that would take some time to determine. And so biochemists find easier ways to study complicated things. And one of the easier ways is through the use of substances that change color. It's a very common thing to do in biochemistry. This is an artificial substrate on the upper left. Okay, this long mouthful of a molecule name. You don't need to know the name. But it's an artificial substrate. And this artificial substrate looks enough like a polypeptide that chymotrypsin can bind to and cut. The chymotrypsin, in fact, does bind to it and does cleave a bond. Okay? It actually cleaves a bond right there. Doesn't matter how or why, just that in fact that it does cleave a bond right where I show you. The result of cleaving that bond by chymotrypsin results in the production of this molecule on the left and another molecule that has a color, whereas this starting material didn't have a color. The beauty of this very simple setup is that one can follow the progress of a reaction by monitoring the appearance of color. And this is something we can do very easily with a spectrophotometer, which measures absorbance of light at specific wavelengths. So we can say how much yellow color is being produced and how rapidly, and we can follow this reaction in essentially real time as it is occurring. Okay? Well, this turns out to be very useful because when we study the reaction, we discover something unusual that happens in the chymotrypsin reaction. It goes through what we call two phases, a fast phase and a slow phase. Okay? A fast phase and a slow phase. I want you to plant those two things in your head. In the fast phase, we see very rapid release of color. And then in the slow phase, we see a more gradual release of color. And the reason that we see a more gradual release of color, it turns out, is that the fast phase with this reaction involves two steps, two big steps. The first step involves cleavage of that bond that releases the yellow color. That happens very rapidly. So we see boing, it goes up. Then the release of yellow color slows down. Why does the release of yellow color slow down? That's because the second phase of the reaction takes a while before the enzyme can go back to cleaving and releasing more yellow color. So what we're seeing here is the recovery of the enzyme from the first, essentially the second process. All right? So let me not be cryptic and show you what I'm talking about. Okay? Here's a schematic diagram of chymotrypsin. You can see the active site with the side chain of serine having the OH group right there. Okay? And here is a polypeptide, or we can think of it as the yellow color, whichever you want to think about. It doesn't really matter. And in the process, what happens, here's the yellow color, what happens is the cleavage step in the first process releases the yellow color, and the release of that yellow color results in the other half of the molecule becoming covalently bound to the active site. I'm going to repeat that. The first phase, the cleavage to release the yellow color, results in the other half of the molecule being covalently bound to the active site. Now, based on what I've told you about enzymes, you should know that if we didn't do something else, we would have a really good suicide inhibitor, correct? Because once it's covalently bound to the active site, the enzyme is going to be dead in the water. Well, it turns out that serine proteases, their mechanism of action involves the formation of a transient covalent bond at the active site. Transient. It lasts for a very short period of time, but long enough that we can see it happen. And when we look at the kinetics of this reaction that we saw here, we're actually seeing the slow release of that other half of the molecule from the active site. Okay? So the two phases are a fast mode, cleavage of the yellow thing off, and a slow mode, which is the release of the other half of the molecule from the active site. Now I'm going to show you the steps in this mechanism in a minute. Okay? I'm going to show you the steps of the mechanism in a minute so that you can see in more detail what I'm talking about. Everybody understand there's two steps to this process. Yes, question? Okay, your question is, if there's a whole lot more chymotrypsin in the substrate, when you do an enzymatic reaction, there never is a lot more enzyme than there is substrate. There's always a lot more substrate than there is enzyme. But maybe I didn't understand. Was there... So even if you did it where there was more... Well, if you did where there was more chymotrypsin than there was substrate, you would basically see one thing happen because you would see the initial release, but the release of the rest of the substrate from the active site wouldn't have any increase here because there would be no more of the original substrate to bind. So that's what you would see. Make sense? Yes, question. Garrett.
Is it possible to have enough chymotrypsin that it'll start releasing, like it'll start releasing through the second phase and then start getting the
Okay, so his question is it possible that you can have enough chymotrypsin that it will be releasing in the second phase while it's starting the first phase? Is that what you're saying? That's, in fact, exactly what's happening right here. Because remember, the only way we get more yellow is if we cut more compound. And since these are slowly being released, only a little bit is able to start going through that fast phase again. Understand? So we are seeing a mixture of fast and slow phases going on throughout the rest of it up here. And that'll occur pretty much at a constant rate. Okay, let's take a look now and see what's happening. If we look up close and personal inside the active site of chymotrypsin, and by the way, what I'm going to tell you here about chymotrypsin is essentially true for every serine protease. It's essentially true for every serine protease. Serine proteases all have a common three-dimensional orientation within them of three specific amino acids called the catalytic triad. The catalytic triad. The numbers adjacent to each one that you see here correspond to their position in the primary structure. So this is, aspartic acid is the 102nd amino acid in the primary structure. Histidine is the 57th amino acid in the primary structure, and serine is the 195th. No, you don't need to know those numbers. Okay? But the fact that they're all in close proximity, as you can see here, is evidence of what kind of structure? Tertiary structure, okay? Because we have things that are far apart in primary structure that are geometrically very close to each other in tertiary structure, okay? Within the active site of this enzyme, and by the way, the active site of this enzyme is you can think of as a hollow chamber. We're going to see there's other things in and around that hollow chamber. So the active site of the enzyme is simply the place where this reaction is occurring, and the things that are making this reaction occur are what you can see on the screen. Okay? The catalytic triad consists of aspartic acid, histidine, and serine. And as I said, all serine proteases work in essentially the same way. How do they work? Well, let's imagine that this serine protease is floating around in solution and it binds to a protein. And a protein is going to have a specific amino acid sequence. And if it binds to a protein in a place where it does not recognize the amino acid to cut next to, what's it going to do? That's a tough question. Nothing. It's going to let it go because it's the wrong substrate, right? It only cuts adjacent to certain amino acids. However, let's imagine it binds adjacent to a phenylalanine. That is one of the amino acids recognized by a serine protease. When it binds to a phenylalanine, okay, the enzyme, remember the Koechlin-induced fit model? What's going to happen when it binds to the proper substrate? Slight change in structure, and that slight change in structure is going to bring about catalysis. It doesn't do that slight change in structure if it binds to the wrong substrate. Well, in this case, it's bound to the right substrate. Now this process I'm going to describe to you up here occurs. When the proper substrate binds, let's say the phenylalanine of a polypeptide chain, the side chain of a phenylalanine binds, the geometry between the aspartic acid and the histidine and the serine changes very, very slightly, because the enzyme itself has changed very, very slightly. This carboxyl group over here is full of electrons. It's negatively charged. If we can imagine we put this carboxyl group slightly closer to this histidine, we can imagine that this histidine ring, which is full of electrons, has electrons that will not like the negative charge of the carboxyl group. The electrons will orient themselves so that they're on the other side of the ring. And on the other side of the ring, because things have been brought a little closer together, there's a very conveniently located hydrogen that is the side chain of a serine. Negative attracts positive, negative pulls off positive, and we're left behind with an oxygen with a negative charge. In other words, the hydrogen leaves its electrons behind because it doesn't need the electrons over here. This resulting structure is called an alkoxide ion, and as we will see, an alkoxide ion is very reactive and is what breaks the peptide bond. We're going to see that in just a minute. This only happens if the proper substrate binds. Okay, now here's the part you're going to love. Let's go through the mechanism. I'm going to step you through it, and I'm going to tell you where I think the critical steps in this mechanism are. Okay? Here's the polypeptide that's been bound. It's in pink above. And here's the peptide bond that's getting broken. Everybody see? There's the peptide bond. We go through the process that I just described to you. The geometric change in shape, the pulling off of the proton by the histidine ring, the production of an alkoxide ion, which is a negatively charged oxygen, meaning it has electrons, extra electrons. Those extra electrons seek a nucleus. It's a nucleophile. The nucleus that it seeks is conveniently placed right next to it in the active site. It's this carbonyl of the peptide bond. The binding of the oxygen to the carbonyl produces, a break in the peptide bond right there. So we've already broken it right here. I'm sorry, we're in the process. Sorry, we haven't broken it. We're in the process of breaking it here. We will break it over here. And there's a transient tetrahedral intermediate that's created right here. Now, this transient intermediate is very unstable. It's very unstable. And unstable things in the belly of an enzyme are not good, because unstable things lead to unpredictable chemical reactions that might result in inhibition, suicide inhibition of the enzyme. To prevent this unstable intermediate from reacting with the enzyme, there is a structure within the active site. Remember this cave? There's a structure in the active site called the oxyanion hole. And it performs one function, stabilizing this intermediate so that it can fall apart. It falls apart in predictable ways. The predictable way meaning loss of the peptide bond. It doesn't react with the enzyme excessively, at least not in the ways that you don't want it to. And one of the peptides is released. So let me just step you through here. Binding of the substrate to the active site, production of an alkoxide ion, formation of a transient tetrahedral intermediate that's stabilized by the oxyanion hole, falling apart of the peptide bond. At this point, this guy right here, this part of the polypeptide that we started with, this part is held in place only by a hydrogen bond. That's not sufficient to hold it to the enzyme. It goes away, as you see here. And that, in the reaction I've already shown you, was the yellow color. The yellow color has disappeared. What are we left with? We're left with an enzyme that is now covalently linked to the other half of the polypeptide chain. We've gone through the fast step. Now we're going to get ready to get rid of the slow step. We've got to break this bond between the other half of the polypeptide chain and the side chain of serine, because that's where it's covalently attached. There's good news. The good news is the mechanism for getting rid of it is very similar to what you've already seen. It involves a molecule of water. Molecule of water pops into the active site, and guess what happens? Here's a lot of electrons in this ring. They pull a hydrogen off of water, leaving a nucleophile, this nucleophile being the OH group, which now attacks the same carbon that we saw before. And now it breaks the bond between between the O of the serine side chain and the second polypeptide chain. The second polypeptide chain is released, and we're back where we started. This second step of getting water to do all of these things is slow. It takes longer for it to happen. Okay, now, this usually generates a fair number of questions, so I'm going to stop and ask for questions. Everybody's going, wow, man, do we have to know all this? Well, let me tell you what I'm not going to ask you. I'm not going to ask you to go up and regenerate this figure, because I think that's just a waste of memorizing time. All right? I think you should know the important things that are happening in this process. Would you like me to go through them very briefly? I had a feeling. I've already done it once, but I will do it again. Okay? The important things happening in this process. First, have to have proper substrate bind to the active site. If the wrong structure binds, nothing happens. If the proper structure binds, then there's a slight change in geometry that results in the pulling off of a proton by the histidine ring to create an alkoxide ion. The alkoxide ion is a reactive nucleophile, and it attacks the carbonyl of the peptide bond. That results in the formation of an intermediate that is stabilized by an oxyanion hole so that it can fall apart. It does. The falling apart releases one half of the polypeptide chain. The other half becomes covalently linked to the serine side chain. We've just finished the fast step of the process. took longer to say it than it obviously would take to do it. In the second step of the process, water comes into the active site. It is activated to be a nucleophile by the same histidine that we saw before, and the resulting OH attacks the carbonyl carbon, the very same carbonyl carbon as before. But now the bond it's breaking is that between the carbonyl carbon and the serine side chain. That results in the breaking of that bond, the release of the second polypeptide chain, and the regeneration of the original intermediate. We've now finished the second phase. So what we've done, we've gone through here, and now you see a little bit about how flexibility of an enzyme helps the enzyme to do what it does. We talked about the magic of enzymes and how enzymes work so much faster than chemical catalysts. They're doing this because the enzyme is flexible. It's not a fixed, rigid structure. Okay? There's one other term I want to introduce here. It's not shown on this figure, but I want to tell you what it is. And that is something called the S1 pocket. Okay? What is the S1 pocket? The S1 pocket is another part of the active site. It doesn't catalyze, but what it does is it recognizes the proper substrate. It recognizes and binds to the proper substrate. What you see on the screen are 3 different S1 pockets for 3 different serine proteases. This little pocket, you could imagine, is where the side chain of an amino acid in a polypeptide chain can bind. It's just a little pocket where these things can bind. All right? In this case of chymotrypsin, the pocket is nice and open and fairly hydrophobic. Nice, open, and fairly hydrophobic. It really likes to bind to things that are fairly hydrophobic and even of a decent size. So phenylalanine side chain, which is a benzene ring, fits in here very nicely. And consequently, the enzyme, the S1 pocket signals to the enzyme, I've bound something that's right. Okay? The S1 pocket changes shape slightly, the enzyme changes shape slightly, and everything happens. If the wrong thing binds, like let's say the side chain of an aspartic acid binds in here, there's no change in the pocket, no change in the enzyme, nothing happens. Okay? If we look at the S1 pocket of the enzyme trypsin, trypsin, you may remember, cut next to lysines and arginines, right? What's the charges of the side chains of lysines and arginines at physiological pH? It's positive. And look what's at the bottom of the S1 pocket. A negative attractor. It binds to it, and it's done its thing. Okay? That's very cool. All right? The S1 pocket of the enzyme known as elastase has blocks within it that aren't the same as the blocks there's no blocks in chymotrypsin. This guy will do relatively hydrophobic side chains, but they've got to be very small and short, because these physically block anything bigger from getting in there. So by looking at the S1 pocket, we can tell which portions of a polypeptide are going to be recognized by these different S1 by these serine proteases. Okay. Let's see. I like to show this figure. It shows you, and I forget which one is which, it doesn't really matter for our purposes. It shows you the schematic structure of chymotrypsin and trypsin. And when you look at this guy, you can see that they're not very different from each other overall. The blue traces the red in many places. What we see among proteins are that related structures, okay, enzymes or proteins that have related function will usually have related structures. And these are evolutionarily related to each other. And you can see they're very, very similar. You might expect that they would be different in one region. Where would you expect they would be different? In the S1 pocket, right? They both are serine proteases. They both use serine in the active site. They both have a catalytic triad. They're both going to have an oxyanion hole. But their S1 pockets are probably going to be a little bit different. Okay. Here's a cool analysis of demonstration of the importance of the catalytic triad. Okay? The importance of the catalytic triad. This is a serine protease called subtlysin. You don't need to know that, but it has a catalytic triad just like the other ones. All right? This measures the kcat of different forms of this enzyme. Here's what's called the wild type, meaning unmutated, meaning it has a normal catalytic triad. Here is the kcat of an enzyme if we mutate serine. This is going from, this is on a log scale, folks, a log of about 2 down to a -5. That's 7 orders of magnitude. That's 10 million fold. This guy here that has the serine mutated to an alanine, meaning it won't have that OH in the active site, this guy is 1/10,000,000th as active as the wild type. It tells us serine in the active site, very, very critical for the function of this enzyme. If we mutate histidine to an alanine in the catalytic triad, we see exactly the same thing. The histidine is pretty critical for the enzymatic function of this particular enzyme. If we convert the aspartic acid to an alanine, what we see is that it's a little bit more active. It's only down by about 6 orders of magnitude, and it tells us that the aspartic acid isn't quite as important as the other two, although it's still very important for the overall function. And finally, if we mutate all three and measure the activity, we see that it's essentially the same as either the serine or the histidine, again, reinforcing the fact that the serine and the histidine are essential for the function of this enzyme. Okay, that's a lot of stuff for today. I've got a couple things for you at the end. I have a limerick. The tert alcohol was a fool, turned to ketone in making a fuel. The work is complete. The bond gave up heat as it lost all its family jewels. There's more. On the most recent summer vacation, only one place deserved condemnation. Its dry summer breezes caused too many sneezes when we got to our first dusty nation. And, of course, we have to have a song. It's called Protein Wonderland to the tune of Winter Wonderland. It's a brand new song. Mechanism determines how an enzyme is working. Here are the ways that one elastase breaks a peptide bond up easily. Starting with the binding of the substrate, catalytic triad is the star. Histidine's electron sink reacts to pull a protein from the serines R. Then the alkoxide ion gets electrons a-flying. It makes a big fuss for one nucleus and breaks and makes a bond with carbonyl. Then the process switches in its action. Water comes to free the carbonyl. Loss of protein yields hydroxide ion. Attacking on the peptide bond there still what the enzyme releases. Otherwise, action ceases. The process is done until the S1 binds the substrate, starting up again. Thank you, guys.